Dear students, today we will talk about nervous system. Nervous system includes two principal parts. It's central nervous system or CNS and peripheral nervous system or PNS. Central nervous system includes brain and spinal cord. Peripheral nervous system includes nerves, nerve ganglia, and nerve endings. So CNS includes brain and spinal cord, and they together provide regulation of different internal organs of different parts of our body, and they provide this regulation via peripheral nervous system, via nerves, nerve ganglia, and nerve endings, which end in different parts of our body and send impulses to different structures, and also they receive impulses which are carried via nerves to central nervous system. So peripheral nervous system provides communication between central nervous system and different parts of our body. Brain and spinal cord, they contain almost all perikaryones of our neurons. Their cell bodies are located in the brain, in spinal cord, and their axons and dendrites excite from brain and spinal cord via nerves. So nerves, they contain axons and dendrites of different neurons, and they carry impulses to peripheral parts of our body, and also they carry impulses, efferent impulses to brain and spinal cord. So nerves, they include axons and dendrites of neurons, which cell bodies are present in CNS. And also some neurons, some perikaryones of neurons are present in peripheral nervous system. They form nerve ganglia. Nerve ganglia are groups, small groups of neurons which are located near inner organs, and they provide regulation of functions of those organs. So let's begin from central nervous system. So, as you already know, central nervous system includes brain and spinal cord, and they provide regulation of main essential functions of our body. Brain and spinal cord, they contain two principal parts, two principal kinds of tissue. It's gray matter, here we can see gray matter, and white matter. So histologically they have some difference. Gray matter, it contains perikaryon of neurons. Perikaryones of neurons are present in gray matter only. Also it contains some parts of axons and dendrites which are beginning from neurons, and also it contains glial cells. So perikaryones of neurons and all surrounding structures, they are present in gray matter, in spinal cord and inside the brain. And also we have white matter. What is white matter? White matter, it contains processes of neurons, axons and dendrites and glial cells, but cell bodies of neurons in white matter are absent. So white matter, it's pathways, conductive pathways of brain and spinal cord. And let's look at localization of gray and white matter in brain and spinal cord. In brain, gray matter forms cortex. It covers brain, cerebral hemispheres, it covers cerebellum, its cortex, and also there are some inclusions of gray matter inside brain. They form different nuclei inside the brain, subcortical nuclei. So they are inclusions, groups of perikaryones of neurons inside white matter. And in the spinal cord we have gray matter inside of spinal cord, and white matter surrounds gray matter outside. So we have different pattern of localization of gray and white matter in brain and spinal cord. Let's begin from brain. Brain contains several hemispheres. It contains brainstem, its diencephalon, mesencephalon, pons, medulla oblongata, and cerebellum. Here we can see diencephalon, mesencephalon, pons, and medulla oblongata. They form brainstem. And histologically, brainstem is composed of white matter, and this white matter contains numerous nuclei. Here we can see nuclei of different cranial nerves, of different essential centers of regulation. But histologically, brainstem has simple structure. It has white matter with numerous nuclei inclusions of perikaryones of neurons. But those perikaryones of neurons, they play essential role in regulation of our vital functions. It's histological structure of brainstem. Let's go to next structure. It's cerebellum. Here we can see it. So what is the structure of cerebellum? Here we can see mid-sagittal section of cerebellum, and it has numerous folds. They form surface of cerebellum, and it has cortex as well as cerebral hemispheres, and also it has white matter inside. And here we can see how branched and complex shape is in the cerebellar white matter. Here we can see branched cerebellar white matter, and it's called arbor vitae cerebelli, or tree of life of cerebellum. Here we can see it. And this white matter is covered with cerebellar cortex. Here we can see white matter. It's darkly stained here, and cerebellar cortex covers this, and it forms numerous gyri, which are also known as cerebellar folia. Here we can see them. So let's look at histological structure of cerebellar cortex. Here we can see white matter, it's a branch of white matter, and it's covered with cerebellar cortex, which forms numerous folds. Here we can see them, cerebellar folia. Here we can see, and it's fold of cerebellar cortex. And cerebellar cortex, it has 3 layers. We can distinguish them: first layer, second, and third. And under them we can see white matter. So what are the layers of cerebellar cortex? First one, second, and third. There are 3 layers, and they are called molecular layer, ganglionic layer, and granular layer. Here we can see. So middle layer, ganglionic layer, also is called a layer of Purkinjeer cells. Here we can see their cell bodies, their perikaryones, and these are the biggest cells in the cerebellum, and they are the most important cells of cerebellum. So they are main cells inside the cerebellar cortex, Purkinjeer cells. And they form middle layer of cerebellar cortex, their perikaryones, and this layer is called ganglionic layer. And let's look here. There are Purkinjeer cells, and their cell bodies, their perikaryones, they form ganglionic layer. And here we can see this layer. It's the thinnest layer because it's composed of single layer of cells. The outermost layer is molecular layer. It hasn't numerous cells. There are single cells. There are stellate cells and some others. They are located here. And molecular layer, it contains branched dendritic tree of Purkinjeer cells. The dendritic trees are going to molecular layers, and here they undergo innervation from different sources. So here we can see a molecular layer of cerebellar cortex. And the innermost layer of cerebellar cortex is granular layer. Here we can see numerous granule cells. They have appearance of granules, and that's why this layer is called granular. They are very numerous, those granule cells, and they form granular, the innermost layer of cerebellar cortex. And under this layer we can see it's white matter, and it hasn't so numerous cells because neurons are absent here. There are only glial cells. Here we can see molecular layer. The outermost layer is a ganglionic layer or Purkinjeer cell layer, and granular layer, and deeper we can see white matter. And covering of this, it's pia mater, which is composed of loose connective tissue, and also there are numerous blood vessels which provide blood supply, nutrition of cerebellar cortex. Here we can see blood vessels and pia mater of cerebellar cortex. And here we can see a diagram which shows cerebellar cortex, 3 layers. The outermost is molecular layer, layer of Purkinjeer cells, and granule cell layer or granular layer. And here we can see that the most numerous cells are present in this, the innermost layer. So Purkinjeer cells, they form single layer. Their cell bodies, they are piriform in shape, and their branched dendritic tree goes to the molecular layer. And look here how branched is the dendritic tree of Purkinjeer cells. Their branches are going to the molecular layer, and they are branched in one plane. So in different sections they may have different appearance according to the plane of the section. In the transverse section of the cerebellar volume they have very branched shape, and in the longitudinal sections the dendritic trees are very compact in shape. And Purkinjeer cells, they have single axon which begins from the basal pole of the cell, and it goes through granule cell layer to the ganglia inside the cerebellum. So here we can see main cells, Purkinjeer cells of cerebellum. And let's look at them once else. So here we can see it's a perikaryon of Purkinjeer cell. It has piriform shape. Here we can see perikaryon and very branched dendritic tree, which is branched, which is located in the molecular layer. Here we can see cell bodies or perikaryons, and their branches, their dendrites are going to the molecular layer. And those cells, they have one axon which goes to the granule cell layer and to the white matter. And here we can see a Purkinjeer cell and dendritic tree and neighboring Purkinjeer cells, which together form a middle layer of cerebellar cortex, the most important layer. And here we can see outermost molecular layer with very branched dendritic trees of Purkinjeer cells, layer of Purkinjeer cells or ganglionic layer which contains cell bodies of Purkinjeer cells, and the innermost layer is granular layer which contains granule cells. And let's look at cooperation of different cells inside the cerebellar cortex. Cerebellar cortex has two kinds of efferent fibers, nerve fibers, which bring information to cerebellar cortex. There are Klimbin fibers and mossy fibers. Klimbin fibers, they conduct information to Purkinjeer cells directly. Here we can see Purkinjeer cell. Dendrites of Purkinjeer cell are located in the molecular layer, its perikaryon of this cell and its axon. And Klimbin fibers, they are going from the granular layer. They are beginning in the nuclei of cerebellum. They are going through granular layer. They follow axons of Purkinjeer cells. They are located near cell bodies of Purkinjeer cells, and they achieve molecular layer. And here their branches of Klimbin fibers, they follow branches of dendritic tree of Purkinjeer cells. And here they contact with them and send them information. Here they form synapses in this molecular layer. It's how Klimbin fiber sends information to Purkinjeer cell. So it directly sends information to Purkinjeer cell. And another way of conduction of information is mossy fiber way. Mossy fibers, they also bring information to cerebellar cortex, but they don't achieve molecular layer. Their endings are in the granule cell layer. So they don't achieve Purkinjeer cells directly. They don't contact with them directly. They end in the granular layer. Here they form contacts with dendrites, short dendrites of granule cells. Here we can see granule cells. They are cell bodies and they receive information from mossy fibers. And here they form glomeruli of cerebellum. They are contacts, synapses between mossy fibers and granule cells. They form glomeruli of cerebellum in the granule cell layer. And those granular cells, they have very long axons. They begin from their cell bodies, and axons, they are going to the molecular layer, to the outermost layer. And here they are T-like branched. Here we can see two branches of axon of granular cells. and those branches, they follow gyri, they follow folds of cerebellar cortex. Here you can see there are axons of granule cells, and here they form synaptic contacts with dendrites of Purkinjeer cells. So Purkinjeer cells, they receive information from axons of granule cells. So here we can see indirect conduction of information from mossy fibers to granular cells, and granular cells, they send this information to Purkinjeer cells. And also there are some blocking cells which block conduction of information of this through this ways. There are stellate cells, basket cells, and some others. So here we can see different ways of conduction of information. Here we can see Purkinjeer cell, the most important cell of cerebellar cortex. It's perikaryon. It's located in the Purkinjeer cell layer. Its output of information, its axon of Purkinjeer cell, and dendricity of Purkinjeer cell. So there are two inputs. It's Klimbin fiber. It goes to the molecular layer and conducts information directly to the Purkinjeer cell. And also another input of the information, it's mossy fiber. It goes here. It conducts information to interneuron to granule cell, which sends this information to molecular layer. It's T-like branching of axon of granule cell, and Purkinjeer cell, dendrites of Purkinjeer cell, they receive information from this cell. So there are different blocking cells, for example, Golgi cells. They also receive information from parallel fibers, from axons of granule cells, and they send blocking impulses to glomeruli of cerebellum, and they block conduction of information on this level. Another blocking cells are stellate cells. They also receive information from parallel fibers, and they send blocking impulses to the dendrites of Purkinjeer cells. And basket cells, they receive information from the same source, but their axons send blocking impulses to the perikaryones or axons of Purkinjeer cells. So different points of blocking of impulses. It's needed to regulate blocking and excitement of the cerebellum and work of cerebellum. So, cerebellar Purkinjeer cells, they have received information from two sources, from Klimbin fibers, from mossy fibers. They provide processing of information. Also, they are under blocking regulation of basket cells, stellate cells, and Golgi cells. And after all this processing, they form impulses which run away from cerebellum through axons of Purkinjeer cells. So here we can see cooperation of cells inside the cerebellar cortex. And now let's go to cerebral cortex. Here we can see cerebral hemispheres, and they also are covered with cortex. Here we can see it forms also gyri, and under this we have white matter, which contains conductive pathways. There are axons and dendrites of neurons, but their perikaryones are present in the cortex. And also there are inclusions of gray matter inside white matter as nuclei, subcortical nuclei of brain. They also contain perikaryones of neurons. So let's look at cerebral cortex. What is the structure of this gray matter of cerebrum? Here we can see a slide, a picture which shows main layers of cerebral cortex. Here we can see that cerebral cortex has another structure. It's different with cerebellar cortex. So the outermost covering is pia mater, which is like pia mater of cerebellum, but other layers, own layers of cerebral cortex are other. So cerebral cortex, it has 6 principal layers. The outermost layer is molecular layer, which is has the same name as outermost layer of cerebellar cortex. Here we can see molecular layer. It has different cells with different shape, and they aren't very numerous. They are present in this outermost layer. Next, it's outer granular layer. Here we can see cells which have granular appearance. Also, we have layer of outer pyramidal cells, or also it's called outer pyramidal layer or simply pyramidal layer. Here we can see it. It's the biggest, the thickest layer. Inner granular layer. So we have outer and inner granular layers. And also we have layer of large pyramidal cells. It's also called inner pyramidal layer or ganglionic layer. So outer and inner pyramidal layer, and layer of polymorphic cells which have different shape. Poly, it means numerous, different shape of those cells in the inner layer. So let's look here. The outermost layer, it's molecular. It hasn't numerous cells, as well as layer of polymorphic cells. It's the innermost layer, but those cells, they have very different polymorphic shape. And let's look at 4 inner layers. We have 2 granular layers, outer and inner, and also we have 2 pyramidal layers, outer and inner pyramidal layers. Here we can see them. So granular layers, they have mainly cells which have granular round shape, and they are very small and numerous. Here we can see outer and inner one. And pyramidal layers, they have cells which shape is like pyramids. Here we can see pyramidal cells. And let's compare outer and inner layers. Outer has bigger thickness, but inner it has bigger cells. There are giant pyramidal cells which are known as bed cells. Here we can see the biggest cells in our brain, and they also are described as biggest cells in our organism. Their size is about 120 micrometers, and it's this size is like size of ovum, which diameter is about 120 or 130 micrometers. So pyramidal cells are very big cells, and they are called bed cells. They form 5th layer of cerebral cortex. It's layer of large pyramidal cells. So molecular layer, outer granular layer, layer of outer pyramidal cells or outer pyramidal layer, inner granular layer, layer of large pyramidal cells or inner pyramidal layer or ganglionic layer, and layer of polymorphic cells. There are main layers of cerebral cortex. Molecular layer, it has cells with different shape as well as layer of polymorphic cells, and we have 2 granular layer and 2 pyramidal layers. Its main structure of cerebral cortex. And here we can see appearance of cells of different layers. It's molecular layer, outer granular layer, mainly round or star-like cells, outer pyramidal layer, small pyramidal cells, inner granular layer, also star-like or round cells, inner pyramidal or ganglionic layer or a layer of giant pyramidal cells or bed cells. They are huge. They are alike outer pyramidal cells, but they are much bigger. And multiform layer or layer of polymorphic cells, it has cells with very different shape. It's a structure of different layers of cerebral cortex. And also we have structural and functional unit of cerebral cortex. It's called module. Module is vertical column of all layers of cerebral cortex, which contain all layers and all cells which have same source of innervation. Here we can see efferent fiber, which brings information here, and all cells which receive information from the same source, they form module. So module is column which includes all 6 layers of cerebral cortex, which have common source of efferent innervation. And also we have different kinds of cerebral cortex. In different regions of cerebral cortex we can have different appearance of cortex and different thickness, different layers. So in different parts of our cortex different layers prevail, and the most important layers are granular and pyramidal layers, which result in different classification. So in some parts of our cerebral cortex pyramidal layers prevail. Here we can see a huge thickness of outer and inner pyramidal layers, but granular layers are almost absent. In another regions of our cortex, we can see that granular layers are well developed, but pyramidal layers are almost absent. They are very thin. So, in some cases, we have very thick outer and inner granular layers and thin pyramidal layers. In some cases, we have opposite situation. We have thick, well developed outer and inner pyramidal layers and thin granular layers. And sometimes we have intermediate structure. So we call those types of cortex granular and agranular types. When we have well-developed granular layers, we can call this type of cerebral cortex granular. And opposite situation, when pyramidal cells are well developed and granular cells are almost absent, we call this type of cerebral cortex agranular type. So here we can see a granular type, well-developed pyramidal layers, and granular type, well-developed granular layers of cerebral cortex. And they have some functional differences. So granular type of cerebral cortex is usually located in sensory regions of cerebral cortex, which provide different types of sense. They are present mainly in sensory regions of cerebral cortex. And agranular type of cerebral cortex in functions, it provides motor functions. So it's not sensory, it's motor type of cerebral cortex. Those regions of cerebral cortex which provide control of movement of our body. And also we have intermediate types of cerebral cortex which provide another functions, and they have different thickness of layers of cerebral cortex. But two opposite types are granular, which is sensory, and agranular, which is motor type of cerebral cortex. And now we are going to spinal cord, another organ of the central nervous system, with spinal cord. Spinal cord, it provides communication between brain and peripheral nervous system. Here we can see brain and spinal cord, and its spinal cord on the cross section. So what is the structure of spinal cord? Here we can see spinal cord and its communication with peripheral nervous system. There are roots, two roots, anterior and posterior roots, and they together they form spinal nerves, and inside spinal nerves we have spinal ganglia. It's beginning of the peripheral nervous system, and later we'll talk about this. And here we can see spinal cord on the cross section. Here we can see gray matter of spinal cord inside and white matter outside. So gray matter, it contains perikaryones of neurons and beginnings of their axons and dendrites. And this region, it's white matter, it forms pathways. There are different axons and dendrites of neurons where they provide a communication between different segments of spinal cord and between spinal cord and peripheral nervous system and spinal cord and brain. So here we can see gray matter. It's white matter, and white matter it forms conductive pathways of spinal cord, and also some axons they excite from spinal cord, and some axons they enter in spinal cord and they form roots of spinal cord. So there are also pathways, bundles of axons which enter or excite from spinal cord. And here we can see appearance of spinal cord in histological slide. It's drawing of histological slide. And here we can see gray matter. It's inside inside the gray matter we have central canal of spinal cord, and it's white matter. White matter is covered with pia mater. It's this connective tissue with blood vessels, which provide nutrition of spinal cord. So let's look at the gray matter of spinal cord. It forms horns, anterior horns and posterior horns. Anterior horns in their functions, they are motor, and posterior, they are sensory. Here we can see appearance anterior horns and posterior horns. And in some segments of spinal cord we have also lateral horns. They aren't so well developed, and they belong to autonomic nervous system. So its appearance. And here we can see star-like points inside gray matter. Here we can see, and there are perikaryones of neurons. And perikaryones of neurons, they form nuclei of spinal cord. They are groups, they are elongated, they follow the longitudinal axis of spinal cord, and on the cross sections they have appearance of nuclei, and that's why they are called nuclei. They aren't round, they are elongated, they have cylindric structure, but on the cross sections they are like round. So here we can see nuclei, and also between perikaryones of neurons there are axons and dendrites and glial cells. And here we can see white matter. It doesn't contain perikaryones of neurons, it contains mainly axons and dendrites, and also there are glial cells. It's white matter of spinal cord. And it's microphotograph of spinal cord, central canal, gray matter, anterior horns, posterior horns, lateral horns, and white matter. And spinal cord is covered with pia mater. Here we can see blood vessels, small blood vessels of pia mater, which provide nutrition of spinal cord. So here we can see gray matter. In gray matter we have numerous nuclei, groups of perikaryones of neurons, which provide some specific functions. And white matter, it contains different conductive pathways, bundles of axons and dendrites, which carry impulses from different sources to another sources. And they form different fasciculae. There are conductive pathways of the spinal cord. Here we can see different nuclei. They are formed by perikaryones of neurons of spinal cord. Here we can see them. And they also may be found on histological sections. And white matter, it contains bundles of axons and dendrites, and perikaryones of neurons. Their axons, for example, from anterior horns, they excite from anterior horns via anterior roots, and they are going to different muscles, to different glands of our body. And spinal cord, it receives information from posterior roots, and their fibers, they enter and they are going to the cells of posterior horns. Here we can see a posterior root. It's in part of information. Axons, they bring information here. Nerve cells, perikaryones of nerve cells, they provide processing of information in the posterior roots. They receive information from axons of sensory cells, and then these cells of posterior horns, they provide processing of information, and then they may send this information to brain, to another segments of spinal cord, or they send this information to motor neurons of anterior horns. And cells inside anterior horns, they are motor neurons, which axons excite from spinal cord inside anterior roots. Here we can see anterior roots, they are composed of axons of motor neurons, and they are going, and anterior and posterior roots, they form spinal nerve. And let's look at this diagram, and let's revise three principal functional kinds of neurons, and also let's talk about reflex arc. So here we can see anatomy of spinal cord. Here we can see spinal cord. It's gray matter, anterior horns and posterior horns. Posterior are sensory, anterior are motor. Here we can see posterior root, anterior root, and they together are fused and they form spinal nerve. It's anatomy of this region. And inside posterior roots we have spinal ganglia. So, what cells are present in spinal ganglia? What cells are present in spinal cord? Spinal ganglia are structures of peripheral nervous system which contains perikaryones of sensory neurons. Those sensory neurons, according to their morphological classification, are pseudo-unipolar neurons. Here we can see their cell body, it's round. Here we can see trunk, common trunk of two processes of dendrite and axon of this pseudo-unipolar sensory neuron. Pseudo-unipolar neurons, usually they have very long dendrites. Here we can see dendrite, and dendrites, they receive information from outer environment, from inner environment, from different sources. For example, sensory endings in your fingers which feel something. And those information, for example, if you touch something, those information immediately runs through dendrite of sensory neuron or pseudo-unipolar neuron and goes to the perikaryon of sensory neuron, which is located in spinal ganglion. There are numerous spinal ganglia on two sides of spinal cord. They are located near the spinal cord anatomically. So spinal ganglia, they contain numerous perikaryons of sensory pseudo-unipolar neurons. And all this information goes through spinal nerves, and spinal nerves, their sensory part is dendrites of sensory neurons. This part of spinal nerve. Sensory information goes to sensory neuron. After this, sensory neurons, they send this information, taken information through the axons to spinal cord. Here we can see axons of sensory neuron, and they form posterior roots of spinal cord. And axons of pseudo-unipolar neurons, they enter in the CNS. So sensory neurons are located in the peripheral nervous system in spinal ganglia, not in the central nervous system. Almost all sensory neurons are located in the peripheral nervous system. They are located in ganglia or in other structures. So sensory neurons. Only their axons enter in the CNS inside their posterior roots of spinal cord. Spinal cord has numerous interneurons which are located in the posterior roots, and interneurons, associative neurons, they receive information from sensory neurons, from axons of sensory neurons. They provide processing of this information. So posterior roots, according to their function, they are sensory, but they contain interneurons. Sensory neurons are located outside. They are located in the spinal ganglia. And here we have interneurons. Interneurons, their number may be different. In some reflex arcs, they are absent. In some reflex arcs, there is one interneuron or two and more. They provide processing of information, and after that they may send this information to brain or to another part of spinal cord, or they send this information to the motor neuron. Motor neuron is located in the anterior horn of spinal cord. So anterior horns of spinal cord, they contain motor neurons which form motor nuclei of spinal cord. And brain also can send information through conductive pathways, through interneurons, to motor neurons, which are located mainly in anterior horns of spinal cord. And after that, motor neuron sends information. It has very long axons which excite from anterior horns inside anterior roots and goes to spinal nerve. And axons are very long and they achieve effectors, muscles for example, or glands, and they provide some regulative effect. So here we can see that posterior roots, they are formed by axons of sensory pseudounipolar neurons. Anterior roots, they are formed by axons of motor neurons. And spinal nerve, it contains dendrites of sensory neurons and axons of motor neurons. So spinal nerves, they are mixed. They have sensory part and they have motor part. Two kinds of nerve fibers are present there. And here we can see three basic types of neurons according to their functions: sensory, interneurons, and motor neurons. And here they form reflex arcs. Sensory neurons are located in PNS, interneurons and motor neurons are located in CNS, in spinal cord. So this way of information is called reflex arc. It includes sensory neuron, it includes interneuron, which may be absent, and it includes motor neuron. Some reflex arcs contain only two neurons. For example, reflex arc of knee reflection, it contains only sensory neuron and motor neuron, and interneurons are absent, and this information directly is conducted directly from sensory neuron to motor neuron. But usually reflex arcs, they contain some interneurons, and interneurons, they are the most numerous neurons in CNS. So our brain, it contains mainly interneurons, and motor neurons, they are present mainly in anterior horns of spinal cord. And all other neurons are different kinds of interneurons. They form different ways, they form different networks, which provide very difficult, very complex processing of information. So it's reflex arc, it's principle of functioning of central and peripheral nervous system. And here we can see what is present in peripheral nervous system. In peripheral nervous system, we have axons and dendrites, which form peripheral nerves. We can see nerve endings, receptors or effector nerve endings, and also some cell bodies of some neurons are located in peripheral nervous system. For example, cell bodies of sensory neurons, which form spinal ganglia. It's peripheral nervous system and its central nervous system. It provides processing of information, and peripheral nervous system, it provides conduction of information and communication between CNS and peripheral organs. And let's look at the structure of spinal ganglion. Here we can see a histological slide. It's drawing a histological microphotograph, and it's covered with thin connective tissue capsule, which covers it outside, and inside we have nervous tissue, different nervous tissue structures. So here we can see perikaryones of neurons. They form spinal ganglion, and they are surrounded by glial cells. They are mental gliocytes, which provide protection and nutrition of these pseudounipolar neurons. Here we can see round cell bodies of pseudounipolar neurons, and only nuclei of glial cells are visible. They surround these cells. And also there are bundles of different axons and dendrites of different nerve fibers. Here we can see spinal nerve and two roots. Posterior root, sensory root, it contains spinal ganglion, and anterior root, it's motor root, it doesn't contain spinal ganglion. So this root, it contains axons of motor neurons. They are going from spinal cord, and this contains axons of pseudounipolar neurons in this part. And here we have dendrites of pseudounipolar neurons, which bring information here. And pseudounipolar neurons, they send their axons inside the posterior roots of spinal cord. So it's structure of spinal ganglion. And here we can see perikaryones of pseudounipolar neurons, and they are surrounded with glial cells, mental gliocytes, and it's connective tissue capsule which covers this spinal ganglion. And it's perikaryon of pseudounipolar neuron. Here we can see its trunk, common trunk, where axon and dendrite begin. They have common beginning, And there are mental gliocytes. Here we can see them. They form covering, they surround and cover perikaryon of pseudounipolar neuron. And these axon and dendrite, they are covered with Schwann cells or neurolimocyte. And here we can see perikaryones of neurons. I think you remember this histological slide. It's a drawing of slide which shows us Golgi apparatus in the pseudounipolar neurons of spinal ganglion. Here we can see the cytoplasm, and there are inclusions. This staining reveals membranes of Golgi apparatus. And here we can see also glial cells which surround these perikaryones of neurons, and also bundles of nerve fibers, myelinated nerve fibers. And here we can see cells. It's microphotograph of dorsal or root ganglion or spinal ganglion. Here we can see nuclei. It's cytoplasm of those cells and nuclei of satellite or mental gliocytes, which provide protection. They are glial cells of spinal ganglia. They provide protection, nutrition of these pseudounipolar neurons. And here we can see cell body and processes which have myelinated covering. So you already know principle of the work of somatic reflex arc. Let's repeat, let's revise this. So dendrite of pseudounipolar neuron receives information, for example, from skin, from inner organs, and it carries impulse to the perikaryon of pseudounipolar neuron, which is located in the spinal ganglion. And axon goes through the posterior root in the posterior horns of the spinal cord. Here we have some interneurons, one, two, or more, or sometimes interneurons are absent. So posterior horns, they contain interneurons. And after that, information goes to motor neuron, which is located in the anterior horns. And axon of motor neuron excites from spinal cord and goes to the muscle, for example. For example, when you touch very hot cup of tea, you remove your hand from those hot cup because of reflex. This reflex works very, very fast, and you cannot even realize how it works. It's somatic reflex arc. And also we have autonomic nervous system and autonomic reflex arc. It has same principle, but there are some differences. So first of all, you should know that we have autonomic nerve ganglia, not only spinal nerve ganglia. We have autonomic nerve ganglia which are located near the inner organs. And there are perikaryones of neurons which send their axons directly to some organs. For example, it's gland, and there is autonomic nerve ganglion, and axon of this cell goes to target cells which react to this impulse. So additional ganglion in this regulation, in autonomic regulation. Here we can see dendrites of pseudounipolar neurons, sensory neurons. They receive information from skin, from inner organ. For example, we are eating something and nose, they react to this information and they send impulse to the sensory neuron, and this sensory neuron send impulse through the axon to the lateral horns, additional horns, which contain interneurons. They are present in the lateral horns, which belong to autonomic nervous system. And in the lateral horns we have perikaryones of interneurons, and their axons excite via anterior roots together with axons of motor neurons. So here we have interneurons, and they form preganglionic fibers. Preganglionic fibers, they go to ganglion, autonomic ganglion, and here information goes to the motor neuron, and motor neuron forms axons, which are postganglionic fibers. So we have preganglionic fibers and postganglionic fibers, and between them there is ganglion and conduction of information in this level. So in the somatic reflex arc we have motor neurons which are located in the spinal cord, but in autonomic nerve regulation motor neurons are located near the inner organs inside autonomic ganglia, and they receive information from interneurons which are located in the lateral horns. It's main difference between autonomic reflex arc and somatic reflex arc. And let's look at structure of autonomic ganglion. It's like spinal ganglion. It also has connective tissue capsule and perikaryones of neurons. But neurons are multipolar. They aren't pseudo-unipolar like in spinal ganglia. They are multipolar, and also they have glial cells, and they have connective tissue capsule. And they are located mainly between muscular layers, for example, in hollow organs, in digestive tube. They are located between smooth muscles and they regulate their functions. So here we can see smooth myocytes, and there are some cells of autonomic nervous system, and they form, here we can see small autonomic ganglion. Also we can see here section of the hollow organs. It's a small intestine, it's ureter, and between the muscles we can find group of nerve cells, multipolar nerve cells, and they form autonomic ganglion, which regulates functions of muscles, smooth muscles, involuntary muscles of hollow organs. Also autonomic ganglia, they send information to glands, so it's autonomic regulation of functions of inner organs, and those autonomic ganglia, they are involved in the autonomic reflex arcs. So here we can see how this reflex arc is working and its basis of functioning of central and peripheral nervous system. And now let's look at structure of nerves. We already know there are nerves, and they contain dendrites of sensory neurons and axons of motor neurons. So sensory nerves, they contain dendrites of sensory neurons. Motor nerves, they contain axons of motor neurons. Mixed nerves, they contain both types, and also autonomic nerves, they contain axons of interneurons, autonomic interneurons, and preganglionic fibers, and postganglionic fibers are present in autonomic nerves. So let's look at the histological structure, how they are organized in the nerves. Here we can see perikaryon of neuron and axon, for example, but sometimes there are dendrites in sensory nerves, and they have myelin covering, except autonomic nerves which are formed by unmyelinated nerve fibers. So myelin is present in somatic nervous system, and autonomic nervous system, it has unmyelinated nerve fibers. Myelinated or unmyelinated nerve fibers, and mixed nerves, they may contain both types. And nerve fibers, they form bundles. It's myelin covering or only covering of Schwann cells in unmyelinated nerve fibers, and they form bundles. Inside the bundles, they have very thin layers of loose connective tissue which contains capillaries which provide their nutrition, and it's called endoneurium. Endoneurium is very thin layer of connective tissue inside the bundle of nerve fibers. If you remember muscles, there were endomysium, perimysium, epimysium, and the same layers of loose fibrous connective tissue are present in nerves also. So between distinct nerve fibers, we have thin layers of connective tissue which are called endoneurium. And endoneurium together with myelinated, unmyelinated nerve fibers, they form bundles of fascicles of nerve fibers. Here we can see bundle of nerve fibers, and those bundles, they are surrounded with perineurium. It's thicker layer of loose connective tissue, and it contains blood vessels which provide nutrition of this nerve. So big nerves, they contain some bundles, and entire nerve is covered with epineurium, which surrounds entire nerve. It's also loose fibrous connective tissue, and it may be even denser than usually loose fibrous connective tissue. So in some textbooks you might find information that epineurium may be dense irregular connective tissue which protects big nerves. It contains more collagen fibers to protect underlying nerve fibers. So we have endoneurium, the thinnest layers of connective tissue, perineurium, which surrounds bundles of nerve fibers, and epineurium covers entire nerve. Sensory nerves, they contain only dendrites of sensory cells. Motor nerves, they contain only axons of motor neurons. Autonomic nerves, they contain only unmyelinated nerve fibers which contain preganglionic or postganglionic fibers. And mixed nerves, they may contain different combinations of those fibers. And here we can see bundle of nerve fibers. Between them there are layers of endoneurium. Bundle is covered with perineurium, and entire nerve is covered with epineurium, as well as muscles. If you remember, there was endomysium, perimysium, epimysium. The same, almost the same names are present in the nerves. It's easy to remember. And here we can see nerve fibers. It's a bundle of myelinated nerve fibers, and on the cross section we can see how they are organized in the nerve. It's thin nerve, it has only one bundle of nerve fibers. So here we can see nerve fibers, myelinated, it's myelin covering, and between them there are thin layers of connective tissue. They form endoneurium, and its covering of the nerve, it's epineurium. It's also loose connective tissue. So different parts of central and peripheral nervous system, they provide communication between brain, spinal cord, nerves, and different peripheral parts of nervous system, and also target cells which are under regulative control of central and peripheral nervous system. And this structure, they provide it provides regulation of all functions of our body. Thank you for your attention.